गुड आफ्टरनून गाइस। सो ये आर्टिकल जो है आज हम इंडियन एक्सप्रेस से लिया है हमने, और जो इसका उद्देश्य है बताना वो है कि हम आज जेनेटिक इंजीनियरिंग के बारे में जानेंगे और जो इंडिया ने डेवलप किया है अपनी देसी जीन एडिटिंग टेक्नोलॉजी, जिसको हम बोलते हैं TNBP और ट्रांसपोसन। एंड आई डोंट नो what's the translation of this, but I read it that's way only TNBP. So okay, सो लेट्स स्टार्ट दिस। तो आज हम इसको शुरू करते हैं। यार मैं थोड़ा मिक्स बोलने की कोशिश कर रहा हूँ, मैं हिंदी और इंग्लिश दोनों में रखता हूँ इसको। सो चलिए इसको पढ़ते हैं। इट सेज द न्यू नाउ देसी जीन एडिटिंग टेक्नोलॉजी टू एड चीपर कमर्शियल जी क्रॉप ब्रीडिंग। एज वी नो दैट we have the gene editing technology which was of earlier was it, that is CRISPR, right? CRISPR, Cas9 and Cas12. So these were the two gene editing technologies we have, but it is both from America. It was developed by MIT and Harvard. Okay, and also it was developed by AgriScience of USA. It is mentioned in the article, though I'll let you know later. So these were earlier gene editing technology, but now what was happening in India, like we have these technology, right? But we can't get a license to commercially grow the crop. So we were not getting the license for these. So when we are not getting the license, so it was getting a bit delay in commercial cultivation of rice or any other crop. So now India has now developed its own genome editing tool, that is TNBP, or it's called transposon. What's it? Transposon. Okay. So it's this name of it. So this was of today article, and let me tell you a little difference. What is there is two things, GM and GE. Okay, so what is GM and what is GE? See, it says GM, genetic modification. It's stand for genetic modification, right? And it stands for genetic or genome editing. Okay, so in genetic modification, what we do, we just modify the DNA. Okay, that is how we modify. We add, like, see, this is the DNA structure, right? So in genetic modification, what we do, we add a foreign substance, like this is the foreign substance. Okay, so when we do genetic modification, we add this. Okay, we add this to the DNA structure and it leads to genetic modification. So here a new foreign element is introduced in the body or cells. Okay, and what is genetic editing? Genetic editing, what it does not introduce any foreign substance. Okay, or it's just like the way it is, and it just make small changes in the DNA itself. It does not introduce any foreign body, okay, in the cells. So now let's start this. What is written in the article? So it says that the TNBP or transposon associated protein, what is the new Desi GE technology of India? It says that it is just like mostly widely used CRISPR associated Cas9 and Cas12 protein, which act as molecular scissor. Okay, molecular scissor means it is a molecular scissor. It is that it's like a scissor which just cut a specific part of a DNA and replicate it somewhere else, or just add a foreign substance through cutting the part and introducing a new substance. Okay, it's a molecular scissor. That's why it's called not a scissor. It's a molecular scissor. It's just a very on a micron level. Na, it does its work. Okay, so it says that the new GE system based on TNBP instead of Cas protein. Okay, it now what is CRISPR relies on Cas protein, but our GE system relies on TNBP. Okay, TNBP. So it says that it is highly effective next generation tool. We have developed a highly effective next generation tool for genome editing in plant, and it was developed by Kutubuddin Ali Maula, senior scientist at Indian Council of Agricultural Research, which is in Delhi, and Central Rice Research Institute, which is in Cuttack, Odisha. Okay? And then it says that we need. Then it says that the advantage with the TNPB protein used by Indian Council of Agricultural Research, TNPB protein. What is the advantage? It says that it is very compact. TNPB proteins are very compact. Says they are much smaller. See, their size is 400 and 500 amino acid, while the cash protein, they have a size of 1000 to 1400 amino acids. Okay, so it's a big number. And see what India have developed, 400 to 500. It's a very compact genetic engineering, right? You can see as there. It is very important facts for prelims, so please do remember what I'm telling you. So the article also tells that the Cas9 and Cas12 are a football and TNBP are baseball. It is basically comparing the size, okay? So it has nothing to do with such. With the hypercompact genome editor using our TNBP, it is sourced from an extreme environmental surviving bacteria. I think this article I will let it crop. Okay, so yeah, it says that it is sourced from an extreme environment surviving bacteria which is called Deinococcus radiodurans. See, it is Deinococcus radiodurans. Okay, so the genetically engineered GE differs from the more host plant, leading to mutation or changes in their DNA sequence. It is what I have told you about na? Genetic modification and genetic engineering, both are different, so we will not go on it, as I have already told you. So it also remember one thing, that molecular Caesar is a different, and there is a navigator, or we call it a guide, that is RNA, deoxyribonucleic acid. Okay, it is act as a navigator. What it act as a navigator. Okay, then there's a Caesars make a cut in the DNA to alter or disable. I have already told you. And then in May 2025, the ICAR announced the release of 2 G rice variant. Okay, see, genetic engineering rice, and what we have announced, we have 2 rice varieties, that is of 2 genetically engineered rice varieties. The first was Samba Mahsuri. First was this, and second was Cotondora Sanalu. So this was another variety. Okay, what we have done, that Indian Institute of Rice Research and Indian Institute of Agricultural Research, which is in Delhi, also remember this, and Indian Institute of Rice Research, which is in Hyderabad. Okay, so it says that Indian Institute of Rice Research, what they used the CRISPR-Cas9 and Cas12 for gene editing. And what they did, I think this is not important, that they edited it. It says that it edited cytokinin oxidase 2. Okay, so it's not important, but remember the 2 what they edited: Samba Masuri rice and Cotondora Sanalu variety. Okay, then they say that a major issue hindering the commercial cultivation of the 2 GE varieties is the IP right. You know what is IP? IP is intellectual property. Okay, IP is intellectual property. Intellectual property is that something that you invent. आपने यार कुछ बनाया है, ठीक है, अब आप चाहते हो वो मेरे पास कुछ टाइम तक मैं उस पर अपनी क्या रखूँ, हुकूमत रखूँ या अपने को एक तरफ से वो चीज़ मेरे पास ही होनी चाहिए, तो हम इसके लिए इंटेलेक्चुअल प्रॉपर्टी राइट्स का यूज़ करते हैं। इंटेलेक्चुअल प्रॉपर्टी राइट्स क्या करेगा, हम गवर्नमेंट को अपनी इन्वेंशन बताएंगे और गवर्नमेंट क्या करेगी, हमें उसके ऊपर कुछ एक वो क्या बोलते हैं हम, कुछ हमें गवर्नमेंट हमें देगी उसके ऊपर कुछ राइट्स देगी, जिनको हम एज अ सोर्स ऑफ डॉक्यूमेंट अपने पास रखेंगे और जिनको हम बोलेंगे कि जिनको हम ये जता सकते हैं कि अगर ये प्रॉपर्टी कहीं और, अगर ये इन्वेंशन कहीं और यूज़ होती है तो हम इस पर अपना क्लेम रख सकते हैं। ओके, देन इट सेज दैट वी हैव इन नेगोशिएशन विद कॉर्टेवा and Broad Institute since July 2025, and these are at advanced stage. But Corteva is an agri science institute. Corteva is agri science institute in America. So okay, so and what is the problem? What is the problem here? The problem is not for research and modification. The problem here is the commercial planting. For commercial planting we need license, and the license have to be get from these Corteva Institute and Broad Institute such as MIT and Harvard. So we are not getting the license. Okay, so what we are now doing is we are using the TNPB, our own genetic engineering tool, to develop these rice varieties so that we can grow it on a commercial scale. So this was the article regarding gene editing in Indian Express. So thank you guys. Please make sure whatever I have told you, please remember it efficiently.